welcome. Now, in the last session, we discussed the melting temperature and its dependence on various factors, and then we started to the very important aspect of DNA, that is how to know the sequence of bases, that means how the bases are arranged one after another, which distinguished one DNA from the other DNA. Okay. There are two methods which are adopted, which were adopted when it was discovered in the late 70s. There were two groups of scientists, one is Maxam and Gilbert and other is Sangers, and it is Gilbert who got the Nobel Prize and Sanger got his second Nobel Prize for discovering how to know the sequence of bases. Maxam-Gilbert's method is called a chemical method. That means, he used a chemical reagent to know the sequence of bases. On the other hand, Sanger's dideoxy method is an enzymatic method. He used an enzyme to know the base sequence in a DNA strand. Okay. So, now today we will discuss the Maxam-Gilbert method first. Okay. Now, to start with, for base sequence, what you need to do, because DNA is a very large molecule and with the large system it is not possible to get this sequence done. It will be very difficult. In proteins also, that when you have a large protein, what you did there, that proteins you chop into pieces, and what are these choppers? The choppers are basically the enzymes, and then you isolate those pieces, small pieces, manageable pieces, and then do the sequence, and finally, you do the overlapping sequence and get the full. In DNA also very similar thing is done. Suppose you have a double stranded DNA which is this big. Okay. So, first thing that you have to do, you have to cut the DNA into small pieces. Now, whenever there is talk about this cutting of the DNA into small pieces, then enzymes come, because chemically that is almost impossible, that is very difficult to cut at very precise places, but it is enzymes. Fortunately, there are enzymes which can cleave the DNA at particular points, where there are some sequences which the enzyme recognizes, like chymotrypsin recognizes aromatic amino acids and breaks the peptide bond. Similarly, there are enzymes which can recognize certain sequences. Here it is not one amino acid, in chymotrypsin only one amino acid, but here some sequence of bases it recognizes, and then once it finds that, it will cleave there. Okay. So, these enzymes are called, first of all, they are called endonuclease. Now, it is not peptidase, it is nuclease, because they are breaking nucleic acid. So, nuclease and then endonuclease, they are breaking the nucleic acid inside the chain. Okay. So, endonuclease. Okay. Now, these endonucleases are known as restriction enzymes. They are very interesting. There are more than 100, 200 possibly restriction enzymes available. These restriction enzymes are there. What they did, whenever this restriction enzyme which is called ALU1, there are different names, BAMH3, then ECOR-I1, so ECOR1 or something, and then this is ALU1. Okay. So, this enzyme, what it does, it cleaves here and there. That means, it cuts the whole thing like this. This is what is called a blunt cut, means you straight away cut the whole thing, because there are different types of cuts possible. One is that you can cut like this, that means this point is here, that point is there, and this is a bigger one and that is the smaller one. This is called that is the blunt cut, and this is the sticky cut. So, all these cuts are possible, but anyway you here ALU1 does what is called a blunt cut, with straight cut. Okay. So, it cuts it into pieces. So, there are two DNA pieces now you have here and there. Suppose you are interested to know the sequence of bases here. Now, what it also says something, this restriction enzyme ALU1 recognizes this AGCT sequence. Wherever there is AGCT sequence, it recognizes that and does a blunt cut between G and C. Now, interestingly, this AGCT, if you look at the complementary strand, it is the same AGCT, because you have to read from the 5 prime end. This is the 5 prime end now, and this is the 3 prime end. So, you are reading from here AGCT, you are reading from there AGCT. This is what is called palindromic sequence. So, all these restriction enzymes, palindromic sequence. So, they recognize palindromic sequence, different palindromic sequences are available. So, different restriction enzymes are available. So, here you are using this ALU1, which recognizes AGCT, okay, which is a palindromic sequence of 4 bases. And so, there is a blunt cut. So, now you have a manageable DNA double helix. Okay. Now, as I said, before you proceed, you have to put a label, a reporter system into this molecule in order to identify later when you do the gel electrophoresis. So, what you do here, there is this 5 prime which is free, because it starts from 5 prime, and here it is the 3 prime. You put there is an enzyme called some kinase enzyme. Kinase enzymes are the enzymes which puts phosphate at the wedge, which does phosphorylation of the free wedge, and there are different types of kinase. Some kinase put does phosphorylation only on the 5 prime wedge and do not touch the 3 prime wedge, whereas some kinase may be there which will do 3 prime wedge phosphorylation and not the 5 prime wedge. Now, here what Maxam-Gilbert did, they took a kinase which does phosphorylation of the 5 prime wedge. This phosphorylation agent is nothing but ATP. ATP is a triphosphate, where one of the phosphorus is labeled with the gamma phosphate is labeled with the radioactive phosphorus, which is called P32, where the atomic mass number is 32. Okay. So, that is radioactive. So, now if you use that kinase and add the ATP with a gamma radioactive phosphate, so that phosphate will be attached to the 5 prime end of the DNA. So, this end will now be attached to the 32 p DNA. Why only this end? Because this kinase is specific for this 5 prime wedge. Enzymes are very specific, it will not go to the 3 prime end. So, obviously, this 5 prime end is also 32 p, which is not written here. So, now you have this piece where the DNA is now labeled with 32 p phosphorus, which is radioactive phosphorus, and you have this double helix. Now, what you do? You heat it and separate the strands, and you isolate only one strand, one of these strands 32 p, because you know these are complementary strands. So, if you know the sequence of one, you know the sequence of the other. So, you have this CT, I am not writing everything because of time, you do not. So, you have this CT up to A C. So, now you have isolated a DNA strand which is now labeled with 32 phosphorus at the 5 prime end. Okay. This is the starting point of Maxam-Gilbert sequence. Okay. Now, this is now, suppose this is my ultimate DNA strand that I will have, a 32 phosphorus here at the 5 prime end, and suppose we take some arbitrary sequence G A T C A G C G A T. I do not know the sequence. I want to develop a method by which I know the sequence of bases here. Okay. Now, Maxam and Gilbert, they were, because I told you it was very difficult to find reaction conditions which will break only between, which will only recognize G and then break the nucleode, that phosphodiesterase bond between the two bases, okay, base sugar. And so, it was very difficult, but they were so brilliant that they could find out conditions, reaction conditions by which they can selectively cleave these bases. So, what they did, they developed 4 cleavage conditions, 4 cleavage conditions. Okay. One condition where, maybe next slide, in one condition where wherever there is A and wherever there is G, then you get a cleavage. So, suppose you have a sequence like this C A T G C A G, and this is your 32 labeled phosphorus here. Okay. And that means, if you utilize this condition, I will come back to this condition. So, what will happen, that wherever there is A, that will be cleaved. Now, here A is here, the question is which side of A will be cleaved, that is a question. In protein, you know, it is the chymotrypsin always does the, at the carboxy, the carbonyl side will break, not on the left side of phenylalanine, but here the reactions are such that it will actually cleave both sides of A, both sides of A. So, if both sides of A are cleaved, so what you will get? You will get 32 phosphorus and C, that will be the piece you will get, and you will get the other piece, that is TGACAG. Okay. But this piece, interestingly, does not have the radioactive phosphorus in it. So, this will not be visible. So, the part on the right side you can neglect, because that will not be visible in the electrophoresis shell. So, we are only interested in the pieces which contains radioactive phosphorus, but this may not be the only one. If there is another A, so you can get a breakage here on both sides of A. If that happens, then you will get 32 P, then A T G C. So, this is A plus G, that means again you have to see where are the G's and then where are the cleavage products. Okay. So, this is one cleavage product you will get, another cleavage product you will get, and then you will also get the cleavage products from the cleavage of G. That means, you will get C, then A, then T, then there will be cleavage of all these G product G. Again I repeat, on the right side I am not bothered, because right side whatever is whatever will release, that will not have any radioactive phosphorus. So, you can discount that. Okay. It will not show up in the gel. What will show up is this piece P phosphorus, then C A T, both sides of G are cleaved, but there is another G here. Okay. So, both sides of G are cleaved. So, you will get, because you will get 32 P and then A T G C A. So, these are the 4 pieces that you will get if you adapt this condition. That means, you subject this piece of DNA and then add DMS is dimethyl sulfate, and then you add dilute H plus, usually formic acid, and then aqueous piperidine. So, if you subject their DNA to this condition, what you will see that there will be cleavage wherever there is A in the chain and wherever there is G in the strand, okay, chain or strand. Then they developed another condition where only G will be cleaved, and the condition is that they just drop this dilute H plus treatment. So, if you take dimethyl sulfate and then aqueous piperidine, then you will get which piece? You will get only the cleavage of the G, that means you will only get this and this, you will not get these two pieces, because there is no cleavage involving A. Okay. And then they got another two conditions, one condition which cleaves both C and T. So, wherever there is C, so we will cleave on both sides of C. So, you will get accordingly the number of cleavage products containing radioactive phosphorus, and they developed also a C specific cleavage, which is hydrazine, then aqueous sodium chloride. So, that reaction only cleaves, and then aqueous piperidine only C. So, basically what they have done? They have identified 4 reaction conditions. In one reaction condition, both A and G are cleaved. Wherever there is A, wherever there is G, that will be cleaved. In another condition, only G's are cleaved. Wherever there is G, that will be cleaved and you will get the piece. In another one is C plus T. So, you get C and T both cleaved. Wherever there is C or wherever there is T, you will get that. And the fourth condition, wherever there is C, that will only be cleaved. So, these are the conditions. Okay. Now, let me give you an example that will I think that will give you much better concept. So, suppose we started with this 5 prime 32p phosphorus, then GATCAGCGAT. I do not know what was the sequence, but I adapt those 4 conditions, or I subject the DNA to these 4 conditions. Basically what I do, I take small tubes which are called Eppendorf tubes, and I take the DNA in every Eppendorf tube, and in one tube I subject that to a condition where both A plus G will be your cleaved. Another is only G specific condition, another is C plus T, and the fourth one is C specific. Okay. So, let us inspect this. Okay. See, this will, if I do the first reaction, this is the number 1 reaction, this is the number 2, this is number 3, this is number 4. So, if I do the number 1 reaction, how many pieces I will get? In number 1, both A and G are cleaved. So, you have to see where are these. First, let us consider A. So, there is a A here. So, I will get 32p and then G, that is my first one. The second one is there is another G. So, I will get 32p G A T C A. Now, one point I missed, and that is a very valid point, that what I am considering here, that one piece of DNA gets attacked only by one molecule of the, or once it gets attacked only once by the reagent. That means, if I have this molecule, you will not get in the same molecule cleavage of both the G's, because the concentration of the reagents are kept at such a level that that statistically what happens, that only one reaction is going to happen to one molecule. So, that is why what we are doing, that we are getting this piece, because if this gets attacked by the reagent, so this will be broken, there is another G and that is in a separate molecule, that G will be cleaved. That is why we are getting 2 different oligonucleotides. Okay. Now, because it is a plus G reaction, so there will be more number of pieces will get 32p, then you will get G. We have considered A now, no? we have considered this is G, and we have sorry, this should be, we are considering only the, so A must be removed. We have first considered the A's. Again I repeat, A's are cleaved, so you get 32G PG, and this A is cleaved, you get GATC, but there is another A here. So you will get GATC A, sorry, A G C G. So these are the three pieces from cleavage of A. Now we have to see what are the cleavage products of G. So if the G is cleaved on both sides, basically there is no nucleotide here. So you can neglect the first one. The second one is this G. So you will get what? 32p G A T C A. That's all. But there is one more G here. So you will get 32p G A T C A, and then G and C. So this is the number of cleavage products containing radioactive phosphorus. Again I repeat, containing radioactive phosphorus that you will get in the first test tube. Okay. The second test tube, where it is only G specific, what you will get? You will get only this one and that one, because these are only the. So second one you will get what? You will get 32p, then G A T C A, and then 32p G A T C A G C. So these are the two from the second test tube. And from the third test tube, if you repeat that from the third test tube, C plus T. So you will get 32p G A T 1, and then you will get 32p G A T. If there is another C, you have to consider C A G. So you will get up to G. You will get. So you will get two pieces for C, and you will get, there are two T's. So you will get two pieces from T. And then from C specific reaction, you will get again those two, there are two C's. So GAT and then GATCAG. Okay. Now we are not writing that. What is important is, without going into that details, we can summarize it very easily, that if you take about this number 1 test tube where A and G are cleaved. So the first, the number of pieces you are getting, but you see the number of bases present in these number of bases present in these pieces. So this contains only one base, this contains 4 bases, this contains 1, 2, 3, 4, 5, 6, 7, 8 bases. This contains 1, 2, 3, 4, 5 bases, and this contains 1, 2, 3, 4, 5, 6, 7 bases. Okay. And in the second test tube, you will have what? You will have 5, and you will have 1, 2, 3, 4, 5, 6, 7. You will have 7. Okay. So now if you do an electrophoresis, forget about this diagram. If you do an electrophoresis, okay, this is your first test tube you are adding here. So how many bands you will see in the because when you take the radioactive photograph, you will see number 1 band, number 4 band, number 8. These are basically proportional to the molecular weight of the whole nucleotide. The more the number of bases, the more is the molecular weight. So your number 1 will be here, then that will be the first test moving, then you have number 4 here, you have 5 here, that will be little bit slower, and you have 7 here and you have 8 here. So, these are the bands. So, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So, that will be the case. And if you take the second test tube, how many bands you will see? 5 and 7. So, that means 1, this is 4, that is 5. So, you will see a band here and you will see a band here. Okay. Similarly, when you take the number 3 test tube, how many bands you will see? That means C here. So, you will get a number of the number of your bases are 3. So, you will get a band between above 4. So, it because it is 3 base. So, that will move little. So, not here, sorry. So, what you do? You this you will not get. This you will get in the third lane. This you will get in the third lane. So, there will be a band here, that is due to GAT, because it is a C specific cleavage, and there is a T specific cleavage also, because it is C plus T. So, you will get 2 also. So, you will get another one here, and then if there is any other C, yes, there is another C here. So, you will get 1, 2, 3, 4, 5, 6. So, you will get a 6 here. Where is 6? 6 is somewhere here, and then you will get there is no other C, but there is a T here. So, you will get 1, 2, 3, 4, 5, 6, 7, 8, 9. So, you will get a 9, that is the slowest one. So, 9 here, and when you have only C, you will get only 2 bands, there are 2 C's. So, 2 C's means one is this 3 you will get. So, 3 is somewhere here, and you will get another C which is here. So, you will get 1, 2, 3, 4, 5, 6. So, 6 we have already said. Where is 6? 6 is somewhere here. So, this is the picture of the gel. So, that is the number 1 lane, this is the number 2 lane, this is the number 3 lane, and that is the number 4 lane. So, now what you do? This is your A plus G lane, and this is your G lane. Okay. So, if it is only A, this you want to know whether this is only due to A or G. If this is only due to A, so in the G lane you are not going to see any. If it is due to G, then you will see both in A and A plus G and G lane. Okay. Similarly, for C plus T, if it is only T, then you will only see in the C plus T lane, but you will not see that in the C lane. But if you see both, that means that it is actually C. So, that means where there are doubling, so this is C, this is C, and this is your G and this is your G, because they are present in both A plus G and G. Now, what you do? You read this gel from this side. So, you can write that first one is A, the second one is which one? This one. This one must be T, because you are not seeing it in the C lane. The third one must be this one, this is your C, because you are seeing in both, and then this is again I repeat, because this needs careful screening. So, A, then T, then C, then A. Let us see whether it is matching. A, then T, then C, then A. We are not getting the first one. First one you have to actually, for Maxam-Gilbert you do not get the first one, because there is no when G is cleaved, you do not get any nucleotide attached to the radioactive phosphorus. So, first one you forget, and then you read A T C A, T C A, then G, then it will be C, because both the bands are present, then it will be G. I am just that way you can continue. So, let us see whether it is matching A T C A G, A T C A G, then C G, and if you continue you will get the other two A T. So, that is how it is done. Okay. it's not very difficult, but the problem with Maxam-Gilbert is that it is very, you need good pair of hands. It needs lot of craftsmanship, no? Because it's a I am not giving you the actual conditions, that how many minutes you have to heat with hydrazine, how many minutes you have to treat with dimethyl sulfate, how many minutes you add sodium your pyridine, aqueous pyridine. So, it is very it is very basic, person dependent. Okay. So, your hands have to be extremely scrupulous. So, that is one problem. Otherwise, Maxam-Gilbert, when it was discovered, it got wide attention, and as you see that Gilbert received the Nobel Prize. Okay. But this is a chemical method. He developed chemical conditions by which you can break different break the DNA strand at different, depending on the base that is present. Okay. I hope this whole thing is clear. Sometimes problems may be given in the other way around, that without giving this one, we give the gel picture, and then from the gel picture you may be asked that what is the sequence of the DNA. But reading the sequence is extremely easy, You just read from the bottom and go further up, and you get from the 5 prime to 3 prime, that sequence you will get. Okay. Now, the question is, there is some chemistry part in it. The chemistry part is, I think this is the same, that why there is specific cleavage, or how was this cleavage of DNA strand. I said both the strand, if you take a base from both side, it is cleaved, both the phosphodiesterase bonds on the 3 prime end and the 5 prime end, both are cleaved. So, some mechanisms are there in the last 2 slides, that this is with dimethyl sulfate. If you add dimethyl sulfate, then only remember the pyrimidine ones are like this. See, now the chemistry part, the pyrimidine ones are like this. In pyrimidine, if you look at the nitrogens, they are not that basic, because they are attached to this carbonyl, they are not very basic. But in the purine, forget about these nitrogens, but this is the nitrogen which is extremely basic, because of the flow of electrons like this. So, this nitrogen is very basic, and dimethyl sulfate is an alkylating agent. So, whenever you add an alkylating agent, wherever there is nucleophilic nitrogen, very basic, very nucleophilic nitrogen, that is going to attack the dimethyl sulfate, and it will undergo methylation. Okay, now, it is a general rule. I am not going into the detailed mechanism, but we can simplify it, that if you have, because this is the general structure of a DNA, this is the phosphate. Without writing phosphate, if I made a circle, I am showing it as a phosphate. So, all the time writing also takes a lot of time. So, you can do it like this. Okay. One thing you remember, that this base, if you can somehow put a positive charge on this base, then what happens, this glycoside bond becomes very weak. This nitrogen lone pair flies here, and the whole base is gone. So, you can actually, basically this is called depurination or depyrimidation, because you are removing that purine base or the pyrimidine base. Okay, but remember, to do that, what you need is to develop a positive charge, and actually Maxam and Gilbert, this is the detailed mechanism that will be given in the slide, but the whole concept is based on this. So, when they did dimethyl sulfate, so this nitrogen is alkylated, that means the whole DNA is at this base has got positive charge. So, if you see the next step immediately your aqueous water comes, attacks here, but that before that this lone pair flies and breaks it down, then water comes and attacks here. So, your base is gone, and it becomes ultimately an open chain. You know, sugars can be open chain also. So, it becomes an aldehyde and alcohol. This aldehyde reacts with piperidine. So, you get this piperidinium. So, that means a ship's base, and this ship's base now what happens, because of this positive charge, these hydrogens at the 2' position become very acidic. So, now there is a beta elimination. Beta elimination means catastrophic, that means this phosphodiester is now leaves. That means, whatever is in the bottom, whatever DNA chain is at the bottom, that goes off. Okay. And as soon as there is a double bond here, so this hydrogen becomes acidic, and that also do the same thing, kicks out the phosphate. So, from the 5' end you have a breakage of phosphate, and from the 3' end also you have breakage of phosphate. That is why I always remember that in Maxam-Gilbert I said that there is breakage from both sides, from the 3' as well as from the 5'. And by carefully adjusting the conditions, they could successfully have developed a condition where only G is affected and A plus G is affected. They knew that if they do not need to only A specific reaction, what they need is A plus G and G, or A plus G and E, that will give you the information. So, they are clever enough to do that, and then C plus T and C. So, that is one mechanism which is given, and the other mechanism is that hydrazine. You can remove the pyrimidine bases by putting the hydrazine, and this is given in, I think that reference is given. You can go to this Voigt and Voigt biochemistry and this mechanism of hydrazinolysis. This is called hydrazinolysis because you are breaking by hydrazine, and that the whole mechanism. Ultimately, this is very similar like the earlier one. Ultimately, you get the Stevens base, this increases the hydrogen acidity that undergoes beta elimination, and there is another beta elimination here. So, you remove both the 3' and the 5'. So, that ends the Maxam-Gilbert. Remember, it is a chemical method, and there are 4 reactions that has to be done, and then in the gel there are 4 lanes, and the species runs according to the number of bases, that means their molecular weight. One important point is the first base that is attached at the 5' that cannot be located or that cannot be determined, because when there is a cleavage there is no nucleotide which has got the radioactive phosphorus. It is only the radioactive phosphorus that is released. So, that is not a nucleotide. So, you will not be able to see that. So, that is the next session we will do the Sanger's method. Okay. Thank you.